Welcome to Spring LIB.
Inheritance, and in particular flies or Drosophila. And here we have a capital S for a straight wing, a little s for a curved wing, big R for red eyes, and little r for brown eyes. Okay, so part A just wants us to very simply use these letters to represent the parental genotypes. So in the first cross, we're doing to true breeding. Tells us that in the first sentence. So that would be true breeding means homozygous for either the dominant trait or homozygous for the recessive trait. So this would be the parent cross. And then, of course, this gives us the standard F1 generation that is heterozygous. And so then we make a test cross with this generation and another set of true breeding recessive. Okay, so part A was just to construct these, and now B is we're going to do our standard Punnett square. So if we have this heterozygous, that can break down into 4 possible gametes, sort of one of every flavor. I'm hoping this little tick on the S helps separate the capital, the dominant, versus the recessive, the lowercase. I hate using S's because they look relatively the same, big or small. Okay, and then, but the gametes for the recessive, we only really have one option. I mean, technically it would make 4 gametes with the exact same recessive in each, but we don't need to make 4 columns, and so we'll do our square here. And we get a heterozygous for both. And then the heterozygous in one, but recessive in the other. And then homozygous recessive. Okay, so this would be B, and then C is just to sort of say, well, what are these genotypes and phenotypes, right? So the distribution here is 25% of each for both the genotypes and phenotypes. Phenotypically, though, this first row would be the straight and straight-winged and red-eyed. Then we have the straight-winged because it has the dominant S. But this time the brown eyes. Then we get into the curved wings. We're at recessive, double recessive for S. So the next 2 will both be curved wings. And then we see the dominant eye color. So for this third row will be red eyes, and then the last row recessive in both. We will have curved wings and brown eyes. Okay, and so that's hopefully had some practice doing things in the standard way there. But then we get to D, and we want to mix it up a little bit. So D is telling us, as it happens, these are on the same chromosome, which means they are linked. So now we do a different cross. And I tend to write my linked crosses a little bit differently. I like to just group them together. So that would mean that our first parent, the very first cross with the true breeding, would, oops, I forgot the ticks here, would look like this. And so then I group these linked traits like this. So then the only kind of offspring ends up being this linked dominant with a linked recessive set. And of course, this problem then wanted us to cross this with another true breeding recessive. Oops, I put a tick on that one where it doesn't belong. So this is the recessive. Okay, and so, but when they're linked, then we only really get two options, right? So because this is the same no matter which way it goes, we either get a combination with the first pair or with the second pair. So the only offspring here end up being either heterozygous in both or homozygous in both. And so that would mean we would maybe expect, without crossover, 50% of each, and the phenotypes would of course be 50% straight-winged and red-eyed for this one that's heterozygous, and then curved wing and brown-eyed for the homozygous recessive. Okay, but E, as we move on, wants us to suppose that 500 flies are produced in a test cross and to apply math methods to calculate the expected number. So if we look at the map that we're given, we can see that the wings, the gene for wings, occurs at 75.5 map units, and then the one for eyes occurs at 104.5 map units. So the difference here is 29 map units. Now a recombination frequency, so one map unit is going to be equal to a 0.01 recombination frequency. So that means these are having a recombination occur 29% of the time. Okay, so we initially predicted that we would have 50% would be this heterozygous, and that 50% would be homozygous recessive. But since 29% are going to recombinate, then we know that 71% will be of this kind. Okay, which means that's going to be 35.5% each. So if we take 500 and figure out what 35.5% of that is, that means we'll have approximately, I'm getting 177.5, so we can say 170, can't really have half a fly, we'll run down. 177 of both of these genotypes. Okay, and so that leaves 29% will be the other two, the recombinated ones. So that's when we have heterozygous in one, but not the other. Or it's also 14.5% each. So if we take 500 and multiply that by 14.5%, that means approximately 72 flies each will be of the heterozygous in one and not the other. And if you add these all together, due to rounding, we're 2 flies short. So maybe we could add 2 to the dominant or the recombinated ones. Doesn't really matter. But that concludes this slightly lengthy multi-part problem.